So SWIFT sequencing is a new method that we've developed at Dana-Farber, which utilizes single-cell RNA sequencing of circulating tumor cells present in the blood. And in this study, we're using SWIFT sequencing to look at CTCs from patients with MGUS, smoldering, and newly diagnosed myeloma, specifically because MGUS and smoldering patients have very few CTCs present in their blood. So we need to find very new and innovative strategies in order to profile these very few cells that are present in the blood. So SWIFT sequencing essentially will allow us to enumerate the CTCs that are present in the blood and quantify the number of CTCs in the blood. It will allow us to look at the proliferation rate of CTCs and in the blood as well. It will allow us to look at the cytogenetic profile and the copy number profile of the tumor cells present in the blood. And lastly, it will allow us to examine gene expression profiles that we know are prognostic in the blood as well. So we've developed a new signature called the circulatory capacity signature, which is able to measure which cells are capable of spreading and moving around the body and essentially metastasizing around the body. They're derived from a CTC signature, so we'll be using that within SWIFT sequencing as well. And overall, SWIFT-Seq is able to compile 4 different readouts that would normally require 4 different assays at the clinical level, and ultimately it reduces costs and it allows us to get more comprehensive information that wouldn't be possible with very few cells. So in addition to clinically relevant information, SWIFT-Seq is also able to provide us new biological insights about patients that have variable CTC burden. So when you look at CTC burden within an MGUS group or within a smoldering group, patients have very variable CTC burden. And usually we'll be able to associate that with they have different bone marrow burden in their disease. We see that bone marrow tumor cells and CTCs are only moderately correlated together. So therefore, we looked at what other features may be contributing to variable CTC burden within patients. First, we saw that cytogenetics can cause variable CTC burden across patients. So we saw patients with T414, T1416, and copy number events 1Q and del13 have higher CTC burden in our cohort as well as within the COMPASS cohort. In addition to that, we also see that patients have a high CTC circulatory capacity signature, have variable CTC burden, which we developed, and again, both our cohort as well as the COMPASS cohort. And lastly, we see that patients that have a highly proliferative bone marrow have a higher CTC burden as well. And SWIFT-Seq allowed us to analyze all of these 3 features that are contributing to CTC burden variability in patients within a single test.